which is an antibody-antigen binding affinity prediction model. So I'll start off with an introduction about myself first and pass it to Sid to do the introduction and the background. So my name is Anushriya or Shriya. I'm a final year PhD student in Dr. Guillaume Lamoureux's lab at Rutgers CCIB. And so our lab works mostly on developing these deep learning models to understand protein-protein interactions, or more broadly, biomolecular interactions. Sid will present alongside with me today. He's the co-first author as well on this paper and really developed some of the foundational models and methods that our lab currently still uses and is heavily involved in this project too. Take it away, Sid, for your introduction.
Great, thanks. I also want to quickly mention that Shreya and I are both married, and we're in the same lab, but I'm also now working in industry. I work at Bristol Myers Squibb, and this work does not represent that. This is my PhD work. So just to clarify. And next slide, please. So we've broken the talk into two parts. The first is the background and intuition, where I will explain the model and the concept of how we made this architecture in 2 dimensions, a much simpler example. And then Shreya will show how she applied it in real data with 3D structures, protein structures, specifically to the antibody-antigen class of molecules for docking and binding affinity prediction. So next slide, please. So first, what are biomolecular interactions and what do they mean in our context? So for us, we consider the interaction as a receptor-ligand model, where we have one molecule binding to another, and these molecules that we're considering are what we consider fully folded protein structures. Next animation, please. So here's a quick animation of some hallucinations from a AlphaFold-like model, and proteins are linear polymers of amino acids that because of their geometry and their sort of charge and other properties, they form locally stable folded structures. So they go from a linear sequence to a structured 3D sequence, 3D structure. And for us, protein-protein interactions are these folded structures that come together to form complexes. And for the SE3Bind project, specifically, it's the antibody-antigen complexes. And so here's an example of proteins performing a complex biological process by forming complexes. Here is an animation of a DNA replication loop, and you can see that the proteins come together and stick together, physically interacting to perform a very coordinated and orchestrated complex function. Next, please. Okay. And also, just to touch, we've extended our models to also work for the small molecule interactions. That is, we have this macromolecule protein, and we have some small molecule that goes and binds to this and catalyzes some reaction or has some other downstream effect. And we applied our models to the small molecules, water and ion, but just a small plug that we have tried this, and the work does exist, but it's out of the scope of this talk. Oh, also, just feel free to jump in and ask questions. If in case I missed something or went over a detail, in this format, I'm happy to take questions. So why did we choose? So we made these SE equivariant models. Why do we choose convolutions? What are important about representations that the models are learning, and what is equivariance? So first, a CNN, or a convolutional neural net, is like a neural network that is like, for example, this perceptron. You have some input X, and you have some weights W, and you have some features Y, where the weights are applied to the input, and these weights are updated based on some reward function or cost function, and we get some output features that help the model represent aspects of the data that minimize the cost of the whatever loss function you choose in the model. For images, however, the input is pixels or values that are on a regular grid, and therefore the weights in the convolutional model are also formed in a regular grid. So the first row of images, you see some creature on the left. If you take this kernel, that is the arrangement of these weights in a fixed regular grid arrangement, and you stamp them out, and you do this series of dot product operations, you get on the image on the right, which is some feature map or activation map. And based on the weights inside that kernel, you will get a different activation. It depends on what the weights are. You can hard code these, like in the bottom row. That kernel is a Gaussian kernel. You can see that the values are radially distributed with the highest value in the middle, and they sum to 1. So what the effect of this is a Gaussian blur. You may have used some photo editing software or image editing software, and there's like a brush tool that goes and reduces the intensity of your image and blurs it, that's like this. You have a kernel with fixed weight that you apply onto an image, and it gives you an output feature, an output. And the operation itself is just a stride of dot products. You have this kernel stamping out sections of the image, and it's a series of sums of products between the kernel and the input. And that leads to features. So the pink box you see, for every stamping action, you get one number out. And you can see that there's the image out that comes out has these new values. And the learning that happens in the model is updating the weights in that yellow box. So the weights will update, will give you a new image, and then those that new feature will be somehow useful, hopefully, to your answering your model's task. Next, please. Okay, so so that's why representations are interesting. That's why we chose convolutions, sort of a spatial arrangement. Now, why do we want equivariance? So here's an example of, for example, is regular CNN on the left. You have is character recognition task, where you want to detect the letter A. Here, the filters are, you need many filters to capture the different orientations of the letter A. However, if you use a SE2 equivariant filter, you can only need one kernel or one filter to detect all these orientations of the letter A. So it's a sort of an optimization for the rotation space, at least for the 2D problem. And for the 3D problem, this helps even more because there's sort of the curse of dimensionality. In this lower dimensions, there's lower degrees of freedom, higher dimensions, higher degrees of freedom, more variability, more room for error. Continuing. Okay, so here's a fun example. So these two images are similar, but they are not identical. And if you orient them vertically, you see that the image on the right actually has the eyes and the mouth upside down. So this is a case where we have the inherent bias of gravity and viewing things upright. So therefore, we don't really have a need to detect if the eyes are upside down or not, or the mouth is upside down. It's sort of an optical illusion, but this is a case where, for molecular context, there is no up or down for a molecule. So it helps to have this rotational equivariance in your models. And the same thing applies to the 3D problem. So that's for the SE3 equivariant neural net. So why do we use these models, and how do we get them? We have kernels. I showed those numbers before in a regular grid. Here are those numbers plotted for a 2D kernel example, and you can see there's nice regular patterns and symmetries. This is this is from a mathematics library where they encode symmetry into the kernels, and it allows you to these are what the kernels look like at initialization, and it allows the model to maintain the representation regardless of how the input is rotated. So the animation on the right, you have a test input, and it has some activation for, say, a binding pocket in this 2D shape. And based on rotating so if you take the input, you run it through the model, you get some output feature that activates at the binding pocket. If I rotate the input, the activation, the feature on the right, is also rotated and preserved. There's no distortion. So this is important for, you can reduce your sampling in your model as well. So by construction, CNNs are already translationally equivariant. If I want to do an image detection task, so all of this work is sort of inspired by computer vision. If you imagine you have a filter that detects faces, it doesn't matter if the face is in the top left of the image or the bottom right of the image, you'll still detect the face. But if the face is rotated, the model may not detect it. This is, if you add the rotational equivariance through the symmetric filters, you get rotational equivariance, and now you have a model that can detect faces whether they're anywhere in the image or rotated. And this is also a useful property for having higher rank features. So you can have scalar features, singular values. You can have vector features, features that have magnitude and direction. And because of the way we have set up our architecture, we can safely transform these higher order features, higher rank features, while maintaining physical properties. For example, a scalar might represent volume, a vector might represent some sort of electric interaction. And next slide, please. Okay, so we first made a prototype of this in 2 dimensions using synthetic shapes. So the figure on the left shows how we generated pairs of shapes. We scattered points on a square, and we optimized a concave hull on it. And then we filled those shapes with values. And here the colors don't mean very much, just to distinguish them, but the values are either 1 inside the shape out and 0 outside. Very simple. These are just synthetic shapes. We made this data up. They don't represent proteins. It's just the idea of how can we score shapes and distinguish good interactions or strong interactions or weak interactions. So in the 2D problem, if on the far right you see we have on the top row some strong looking interfaces, and their score that from our data set generation, synthetic data set generation, is more negative. So they have a stronger interaction. And then the bottom row of non-interacting shapes have a lower negative score or less negative score. So they have weaker interactions. So we are able to distinguish that. Next slide, please. So if we want to define what interaction is, we thought the first thing to do is let's have an energy function that describes interaction. So the first thing we did was we decided to have first constraint was we wanted a Boltzmann distribution of transformations. That is, we have a probability for every state of the receptor and ligand. So if
Yes, I'm missing something on the previous slide.
Yeah. Yes.
You have these synthetic shapes and, you have some sort of 2D equivariancy. And we are seeing higher binding free energy, so to say.
Sure, yeah, that's right.
And on the bottom, it is reducing. And what I'm missing is, what is the attribution for this? Like, why is higher for the shapes above and
right. Down? So we have features that we score with a correlation, an FFT correlation. So we have a scoring function that's based on the features of these shapes that, for example, if we have two shapes and they overlap, it'll be penalized. If we have features that describe just the outline of the shape, if they overlap slightly, that'll be rewarded, meaning we want to bring the shapes together, but we don't want them to occupy the same space. So we have a scoring function to just generate these training examples. This is just the training data set. I'll show you the learned model in a few slides.
All right.
If that answers. So this is just like from our synthetic data set, we generated hard-coded features just to define this synthetic universe of shapes and how they interact. And then later we applied to 3D. So it's very abstract, but it works for prototyping, and it helped us solve a more complicated problem. But good question. Thank you. Okay, so we have a Boltzmann distribution describing our transformation. So we have receptor ligand, and based on how the ligand moves around, we have a associated probability. And this is goes to statistical mechanics, and this is a partition function, and where the energy in that partition function is based on the movement of the different positions that the receptor and ligand occupy relative to each other. Next, please. Okay, and then one more constraint is that we ensure that the best transformation has to have the minimum energy. So next animation. So here is a figure of the energy landscape of two shapes. So we have a stationary receptor in black, and we have a mobile ligand in red. Next animation. One more. Okay, so there's a minimum energy rotation and interface forming from this example. So in the next slide, I'll show how we got this scoring, how we got these values. But for every rotation of the red ligand, there is a minimum energy translation, and that is what the bottom red curve shows. There is a minimum energy translation given a specific orientation of the ligand. And there are competing poses in this example. So the minimum energy one has the tightest appearing interface, but the other examples have sort of looser interfaces. Next animation. And one more consequence of this is once we generate this energy landscape, using some operations, we can turn it into a free energy. And as Lukman pointed out, that's right. That number we had was effectively like a free energy. So we get a lot from being able to score interactions by parameterizing as energy. Okay, so now how did we get these scores and next animation? So the first thing is we borrowed an idea from a long time ago from Kachalsky-Katzir in 1992. So some very prominent scientists, physicists at the time, they devised that they could solve molecular interaction by just having these kind of shapes describing their molecules, a bulk and a boundary feature for each shape, for each molecule. And just by doing FFT and hard coding these shapes, they could score it. And that's essentially what we've done. And at the time, they didn't have access to modern deep learning or the compute. So they didn't make much progress, but the idea was there. Next animation. So here's how we actually do it. So taking in this example these hard-coded features, we do a pairwise correlation of these feature sets using FFT. And FFT here is just a very fast way of doing element-wise multiplication of the features and the space that they occupy. So remember, these are images, right? So they have values inside their shape, and they have value outside. And we have these A coefficients, A00 all the way to A11. These will score the contribution of each of these pairs of features when we correlate them. So for example, if we take the two zero shapes, the two black shapes at the top, and we were to score them, and during the correlation, there's a, since it's an element-wise multiplication, there is a space where the shapes are overlapping. That will be penalized. And if we want this score to be an energy, then the A coefficient will be a positive value because it penalizes the overlap of these shapes. But then on the bottom, we have these boundaries. If those boundaries start to get close together, we want that to be rewarded. So the A coefficient would be a negative, therefore reducing the energy of this interaction. And it's very abstract, but I'll show it with real examples and what the model has learned next. So given those synthetic shapes with this basic rule of interaction, we had a model learn features independent of those hard-coded features from the input. They had no access to that. The model learned its own features to try to best describe how this 2D space of shapes interacting works. So on the left, we have features that are scalar and vector, and these can describe physical properties. For example, scalar can describe the volume of the shape, or the vector can describe the potential at the surface of the shape. Yes, question? Question?
I think we had lost voice
of
you, but I think I can hear you now.
Okay, I can start the slide again. So learn feature examples from coming out of the model on the left. We have scalar features on the left column that could describe volume of our shape. And these are learned features. And in the right column, we have vector features that could describe potentially electric potentials, things like that. So they have magnitude and direction. So this is, and they have components. So they have X and Y component in 2 dimensions. And there's a spatial interpretation from our correlation. So the red and black shape is an example with the best docked pose, the best scoring pose of these 2 shapes. And on the right is the FFT correlation for that single orientation, rotational orientation of the red shape. So you can imagine if we were sampling in a circle to 360 degrees, right, or from minus pi to positive pi, for each degree increment, there will be a different correlation coming out of it. So we'll have 360 of these correlation slices in the figure on the right. But for this orientation, there is a best pose, and that is it can be interpreted as the distance or the position away from the origin of that correlation on the right. So there is an XY value that describes that is on this energy surface that is this equivalent to the translation of the moving that red ligand away from the center of the so these shapes when we generate them, they're standardized, they're on top of each other. So that purple arrow for the best dock complex is how that ligand translates out. It's the best scoring based on this FFT energy. So the model essentially learns for every orientational ligand and energy, so through FFT correlation, and the minimum energy in this space is the translation that gives us the lowest energy pose. And on the landscape on the bottom, you can see for every rotation, there is a minimum energy translation. But since it's a learned model, this landscape is much more pronounced than the previous landscape I showed. That was the hard-coded feature that had multiple competing scores. At least for this example, and with the learned features, we get a dominating single low energy peak for that one pose. So our model is able to learn how to put together 2-dimensional pieces of synthetic shapes that are supposed to be molecules. And next slide, please. And sort of to help transition Shreya's talk, I guess, the idea is that if you take this model in 2D, right, you have 2D synthetic shapes, and you apply convolutions, you learn features, and you do correlations of those features give you an energy landscape that describe how to bring those 2 shapes together. The same thing applies in 3D, and Shreya, you can take it away.
Yeah, so what Sid did was prototyping in 2D, and we took that working and concepts from 2D and applied it to 3D. And particularly, we look now at the antibody-antigen binding affinity and docking problem. Here on the slide is just a high-level overview of the workflow. So we have our redocking steps, which I'll explain, and we have these feature extractions. And I also examined in this paper the interpretation of some of these features and how we can actually examine what the model is learning. And we do a similar cross-correlation with FFT that Sid described, and then we calculate the binding affinity. So I'll go into detail about how we do this in the 3D problem set. So before I start with that, I'll just go over the data set generation for this project. So we look at antibody-antigen pairs, and the structure at the top is showing the full antibody structure that contains this Fab region, which has these antigen binding sites, and it also has this FC region that's used to bind to the cell membranes. Most PDB files don't have the FC region, and so they contain only antibody-antigen binding arms. So from these various databases, we extracted the complexes, and from the available set of PDB files that we had, we simplified the problem. Instead of looking at antigen bound by 2 antibodies, we only select one antibody-antigen binding pair. Again, this is just to simplify the model a little bit, so we have fewer working pieces. So our data set looks at just the antibody with the heavy and light chain, and then the antigens, which are either monomers or dimers. So we cluster our antibody and antigen data set using this antigen UniRef 50 sequence identity. And this is really to ensure that we are separating our data set enough so that the training and validation sets have non-overlapping examples. And I'll kind of go over the importance of this towards the end. What we also do is when we put our proteins in this grid, we have a size limitation. So we filter out antibody and antigen complexes less than 150 angstroms. This is just to ensure that they fit into our grid size of 75 by 75 by 75. We also make sure that individual monomers involved in that complex are less than 100 angstroms to, again, ensure that they fit properly in the box. The figure on the right is showing the experimental delta G distribution. And we have a handful of examples that have more than one reported delta G value. And for those, we use the average of the delta Gs for the SE3Bind model. The last thing we do for our data set is we run all of our examples through Modeler, and this is to standardize the complex so we can generate missing residues and also standardize the stereochemistry. This also allows us to have some flexibility representation in our rigid body modeling that we do. So we can have some examples with slightly different flexible ends as well. And so our training data set is composed of just homology models, but in our validation, we also look at homology and the crystal structure examples. So taking the ideas from the 2D and applying it to 3D, we follow a very similar approach. So the first thing we do is we place our receptor and ligand or antibody-antigen onto this grid. And similar to how an image has RGB channels, we split the heavy atoms of the protein, so carbon, nitrogen, oxygen, sulfur, into separate channels for our coordinates. We also have this bulk channel that contains all the protein coordinates. We do the same thing for the receptor ligand complex as well. And we place these on a 2 angstrom resolution grid in our box. And so our input contains 5 scalars, which are just the 5 heavy atom channels of our proteins in this manner. And they're passed through the SE3 convnet. So there's a Siamese convnet for receptor and ligand together, and we output these features. So features 1 and 2 are scalar features, and then we also have vector features. I'll talk a little bit about feature 0 separately when we get to the binding affinity task. So the way we split up our task is very similar to how we did it in 2D, where we get an energy grid, energy values, and then we score that and are able to get delta G. So for this project, we train our model in 2 separate tasks. The first one is what we call task 0. So we pre-train the model to do the redocking of the antibody-antigen complexes. So we're getting this energy grid, and from that we can extract the best energy value that corresponds to the best pose. And from that, we can extract delta G values, and that's what task 1 is, which is the binding affinity prediction problem. So I'll go into detail about how we do each of the 2 tasks. So for the redocking problem, these receptor ligands are passed through the conv-nets and they go through the FFT correlation step that Sid described previously. And similar to the 2D data set, we also have this A coefficient. So our energy term comes out to the sum of the correlations of the features and the receptors and the ligand with respect to the translation and the rotation. But as Sid mentioned with 3D task, the curse of dimensionality. And so to simplify the problem, we actually give the proteins in the correct orientation. So the model is really only learning the translation that's required to bring the ligand in complex with the receptor. So the model is only learning translation. Again, this is to simplify the task a little bit. And we have these A coefficients as well. And when we formulate the A coefficient in this manner, where A2 is equal to the minus A1 and A4 is equal to minus A3, this allows us to have a special interpretation that can tie these features to physical properties. So as Sid mentioned, we have these geometric features that are scalar and vector features. These can encode potentially charged features for a scalar vector, and for the vector features, we can have dipole or electrostatic fields because vector features have both direction and magnitude. So we can have some level of interpretability of our features coming out from our conv-net. So the energy term, again, mentioned that we give the receptor and ligand in the correct pose, and the energy that we get out is the ligand translation that minimizes that energy, and that corresponds to the best pose. And this is what we will actually use for our delta G prediction formula. So for task 1, the binding affinity prediction, we can think of it more commonly as the Gibbs free energy that I'm showing at the top here, that has this enthalpic contribution minus the temperature and the entropic contributions. We can also think of it in terms of partition functions as well that I've shown here, where it's basically looking at the number of n number of receptor ligand in the unbound state and the bound states in solution. But we can simplify that a little bit more by assuming that these receptor ligand behave independently. So we can look at just the partition functions of the single copy of fragments, which is what I've shown here. After doing some other derivations, we end up with our final delta G formula, which takes the energy that we learned from the previous task, which was the best translation that gave you the minimum energy for the best pose. And that's what this term is. We also have this delta E naught term, and this is where the feature, just to mention that we do this because we also assume that the bound state is dominated by a single conformation. So the best pose is giving the best bound state. And in this task, we also include this E naught term, which is where feature 0 comes in. So this third scalar feature that we have is meant to capture these entropic contributions. So any other contribution to binding affinity that the docking task doesn't already encapsulate. And this can be thought of as sort of the sum of the bound features minus the receptor and ligand unbound. So this is meant to be a term that captures some of these like entropic contributions, similar to the Gibbs free energy that I mentioned. Lastly, we have this N naught term, and this is defined as the constant representation of R and L if we were to define a delta G in standard state in solution. Another way to think about this term is more intuitively the contribution, the box that we have these proteins in to binding energy. So this is a constant learnable term that accounts for some of these other contributions to binding that are present through our delta E naught term as well as this energy term. So the final binding affinity prediction equation that we come up with is shown here in this red box. Before I move on to results, should I stop for questions if there's any? Because this was a lot of detail.
I had a question here, but okay, let me ask.
Sure.
Because the framework is you're presenting seems to be separating like the direct interaction energy from sort of like additional inbound versus unbound correction. So okay, I see it, but I feel like it trickles down like a parameterization than someone really appreciating it, and how can I really see this in sort of like so being a parameter, but someone appreciating it in sort of an evident way within the experimentation, so to say. I don't know whether that.
I guess I can help with the interpretation then.
Sure.
So
Okay.
whenever you see a delta, it's always like final, like delta v velocity, right? Final velocity minus initial, right? So same thing
Sure.
here with delta g, right? It's final state minus initial state. So the final state is the bound state, right? And the initial state is the unbound state. So you can sort of think of it like that. We're looking at what does it take to go from the unbound state to the bound state, and we're doing this with rigid bodies, and we're breaking up these features into to have we're sort of, like you said, parameterizing. It's like a form of regularization. We have all these features in our neural network, and we're sort of confining them into saying that, okay, these set of features treated this way will be entropic. These set of features treated this way will be enthalpic. Sort of like that.
Sure. At least. Sounds great.
Yeah, okay. I'll keep moving, and maybe if I think of a better way to explain it, I'll come back to it. Yeah.
All right.
Okay, so this is how we set up the task 0 and task 1 for binding affinity prediction. And for the binding affinity task itself, we actually jointly train on the docking task and the delta G prediction task. So the model is seeing both the energy landscape and the delta G calculations in the same data set and the same flow. But we do also pre-train on the docking alone as well. Okay, so I'll move on to some of the results. So the first thing I'll talk about is these interpretability of these scalar features. So as we mentioned, these geometric features like the scalar features and the vector features, and the way we formulated our correlation the energy term, we can have some level of association to physical properties. Like the scalar features can describe volume, so that can describe charge, and the vector features with the magnitude and direction can describe these electric and dipole features. And so in this example, we looked at just the scalar features, and the figure on the right at the very top is the protein complex shown in the electrostatic potential color scheme. So the blue are the positive regions and the red are the negative. And the second row is the antibody on the left, and then the antigen on the right, showing the same thing again. And what we looked at is the sum and the differences of our scalar features. And what we find is, although we don't make direct connection to the features and the physical properties, we can see from the overlapping regions of the blue with the teal show, and then the red and the orange can show that the model is learning to capture these variations of the properties, at least along the entire surface of the protein. So the model is learning some level of charge or some of these electrostatic potentials using just scalar features. And so we don't want to take this into having, a direct connection to features and physical properties, but just a way to explore what the model is learning through these geometric features that we have. So looking at the delta G prediction results.
Sure, on the previous slide, I think it's something interesting there. When you look at the protein app, and you're looking at the scalar features and the vector features, from a basic physics perspective, someone would think that the scalar features are trying to demonstrate something like sort of like a quantity somewhere. So which I think you're putting in quotes charge, and which means it's not real charge and it's not real electrostatic potential. And when you look at the vector features down, I think the vectors should be.
These are all scalar features, actually. We don't look at vector features because it's a lot more difficult dealing with the magnitude and direction chart. So we only look at the scalar features here. So this is the bottom row is the scalar features of the feature 0, the entropic contribution. And then these are the sum and the differences of the two scalar features that we have.
Okay, my bad. So okay, that makes more sense. So if right now you're not focusing on the vector features, because I was thinking the vector features should be giving us some sort of maybe orientation or some sort of direction, and I was like, how can that be viewed? Like, how can we come to realize that? But then finally, still on the scalar features.
Yep.
For someone to really understand and appreciate, if these are not real charges and not dipoles, maybe in a biological point of view, what can be said about these entities that we are trying to abstract with these physics terms?
I
So
can take it. If you
yeah,
want to
if you
answer,
want to try.
okay.
Well,
I
I
would
was just
say
gonna yeah, go ahead and I'll
most
join
simply,
in.
these features are sort of complementary, right? They don't have to be perfect complementary, but at least with like the 2D example, right? There was some shape complementarity that helped bring those examples together with some arbitrary scoring, right? Here, similarly, we don't have a label for the charge in our data set, right? We don't have a label for the electric field, no,
Yeah.
unless we maybe go generate that. So because of that, we have these quotes, and like the interpretation is a little open because we don't have ground truth for these values. So in
So
that sense,
yeah,
but
to add to
I just want to
let
say,
me just say this
okay, go ahead, go
go
ahead,
ahead.
go ahead, go ahead. Okay,
I
I'll
was
go.
just gonna
Okay,
say
okay.
go
Look,
ahead.
good intuition about the vectors. Shreya, you may be able to show what you've been doing with the vector features if there's time.
I was just going to talk about that. So
Okay.
because these features are learned directly from the structures themselves, we don't give any other information, like Sid said, that these locations are charged or not. This is simply what the model has picked up from the input structures that we've given. And so the idea here was mostly to understand these scalar features, which we know should tie into these volume or charge properties. Are they actually showing that? So this was more of a exploratory analysis to understand that a little bit more. So here, in quotes, I say charge and electrostatic potential. Again, it's not direct because we don't have labels for them. And so this is all sort of abstract scalar features that we're trying to assign some meaning and properties of proteins to. So that's why it's a bit more abstract, and that's why we don't make direct connections to these properties. We just know that the model is picking up on signal that we know should associate with some of these charges. In terms of the vector features, actually, so in this project, we didn't deal with vector features, but in my current project that I'm working for my PhD, I do look at vector features, and just based on preliminary results, there does seem to be some notion of association with charged residues. So there is a way to visualize it. It's just not as simple as visualizing a scalar feature. So maybe I can share that next time we're talking. Yeah.
Okay, there's a way I'm obsessed a little bit about this because, but finally, just to clarify on what I'm thinking is what the model is trying to do here. Will I be right if I say it is characterizing the outer sort of structure of the protein, how it is looking? Is that sort of thinking?
So that's actually one of the things is that because of the way the convolutional kernel is moving, it's going inside the protein, it's going outside in the empty space around the protein. It's screening the entire three-dimensional space of essentially what the input is just atom coordinates and
Okay.
as a 3D image. So instead of pixels, right, saying channels of red, green, and blue, you have a 3D image with channels of carbon, nitrogen, oxygen. And when you stack them together, you get a full, like imagine a pi mole visualized or a protein structure, right? So so
Okay.
that's actually one of the advantages of these models is that you can detect buried features. So deep pockets and so on.
So I could be right if I say the model is trying to bring out the sort of latent edges of the protein and features, so to say.
Yeah, exactly. The latent features that are present in the structure that we can extract from the contents. Yeah.
Thank you.
And the architecture is we pre-train the model to dock two proteins together, and from those features we try to extend and predict a binding affinity. So that pre-training task of docking those features helps bring out, essentially that's an interface detection model, right? It detects
Perfect.
interface. Yeah. And
I think you nailed it.
And because the model has rotational equivariance in the filters, you end up just, if you can detect the interface here with these two proteins, a similar interface on another protein in another location might also be detected.
Thank you.
Yeah. These are great questions, by the way.
Yeah. So because this is such an abstract feature, we didn't go too deep into trying to have real interpretation. It's just more the general idea that these models are learning some properties of proteins. Okay. So for our model itself, we did a lot of ablation studies, and one of the, I'll just go over some of the two, I guess, main points. When we look at features 0, which is providing the indirect contribution to binding, we tested two different conditions where we constrain feature 0 to be strictly positive, and we find that it actually performs really worse on delta G predictions, and that can be seen to this MAE, or this mean error values. And when we set it to equal to 0, it doesn't have it doesn't degrade or improve the performance, sort of neutral. We also tested using two different types of loss functions, the L1 versus the L2 regression loss. And what we find is that when we have this L2 loss in our binding delta G prediction task, it actually displays stronger signs of overfitting. Again, you can see that with the MAE values here. So the best model that we chose among our ablation studies that we call SE3Bind is the model that has the least overfitting and the greatest generalization. And so the correlation of the predicted delta G and the experimental delta G is shown on the figure on the right, with the green spread is showing the training data set that has a Pearson correlation of 0.56. And as I mentioned previously, we do validation on two different sets. So we do crystal structure, which is in blue, and the homology models, because that's our training data, which are all homology models. And both of those score fairly similar, 0.24 and 0.26. So we had this model and we saw the correlations were fairly okay. The next logical step was for us to compare SE3Bind to existing binding affinity models. So we took existing models that were evaluated on the same data sets, so AB-Bind, SCEMP, PDB-Bind, and we compared how SE3 performs in comparison to that. So after compiling this data, something that we noticed was that these existing models show really high Pearson correlation scores. So for example, this top net tree has a Pearson score of 0.85, and same with these prospect EL and some of these other models. And so comparing these values to our SE3Bind, which has a training correlation of 0.56, we were curious, how can we explain these models? Like, what is going on with our model versus these existing models to explain the low performance? And I'll put low in quotes. So we dug a little deeper into some of these existing models, how they were set up, what data sets they use, how they were split. And this is sort of where things got a little interesting. These existing models, many of them randomly split training and validation examples. So that means that there are similar examples in both the training and the validation sets. So you just take a bunch of PDB files and you randomly split them and you do your analysis. But that leads to data leakage issues. And that's something that some of these newer papers have been exploring, such as this GeoPPI, which split their data set using this evolutionary classification domain, the ECOD, where they split the protein not just based on the structural homology, but also based on evolutionary classifications that to ensure that their training and validation sets are truly different. And when the authors of GeoPPI retrain this topnet tree using the dataset split by ECOD, the performance drops pretty significantly from 0.85 to 0.38. Similarly, the authors of Graffinity, when they have a randomly split data set, the correlation score is really high, on par with the topnet tree, but they split their data set further in using the CDR sequence identity of the antibodies. And you can see that various cutoff thresholds of CDR identity clusters, the performance drops pretty significantly. And as I mentioned at the very beginning, our SE3Bind dataset was initially split using antigen UNIRA 50 clusters. So we were already aware to separate our dataset, training and validation, to ensure that we had minimal overlaps. And seeing sort of the methods that Graffinity and GPPI were using, we examined further what if our performance can be explained by the dataset. So we followed the paper Graffinity's method and looked at the heavy chain CDR loop sequence identity. Particularly, we look at the H3 region, which is the main determinant of binding specificity. And we find that between our training and validation set, which is this green curve here, the median identity is 20%. And we find that only 3 antibodies in our entire data set share 100% CDR sequence identity between training and validation set. So we have a minor data leakage, but generally our data set contains non-overlapping examples. And so in line with what we saw in the previous table, when we split data set using a stringent structural homologies, your performance actually drops fairly low. So one of the key takeaways from this paper I want to say is that data set splitting strategy is very essential, especially for a structure-based model. And we can see that these overestimation of these existing models is partly due to the random splitting of data set. And so when we take all of that into account, we find that SE3Bind performs comparably to these existing models when we have a stringent data set splitting strategy. And this is particularly important because the antibody-antigen binding affinity data set is already so limited. And we see that when we have this random splitting, the existing models are overfitting on the limited data set that we have. And so the key takeaway is that we need to have a more biologically or structurally informed separation to have a more accurate and generalizable model. And SE3Bind is sort of in a place where when given enough data, we can have more complex volumetric representations. So currently we have 3 scalars and 2 vectors, but we can have more complex representations that can help describe the structural determinants of binding affinity given more data. So this model, when we take these constraints into account, is actually performing fairly well to predict binding affinity. So the main takeaways are that we can use CNNs to give volumetric representations of binding affinity and look at the structural determinants of that, as well as looking at the interpretability of the features that are coming out of the CompNets. And I'll end with just acknowledgement side. So thank you to Dr. Guillaume Lamroux, our advisor, Dr. Sid Bhadra for building and making the foundation models. This work would not have been possible without OARC, Rutgers OARC computational resources, CCIB, and the NSF NRT fellowship funding. This is just an image of our our current members of the lab generated by Gemini. We did not actually go to a theme park.
Okay.
Thank you.
Thank you, Shreya and Sid. I have questions, and
Yes.
I'm also inviting members, if you have any question, you'll just unmute yourself or put it in the chat. Yes, my first question is about the homology concept, and I think you achieved this with the modeler, I think, to cater for the stereochemistry, but also I think for the missing residues. And I'm curious. I want to know. Yes, it's like you're catering for these missing sort of antigen, I think antibody, if I'm right, and within the data set. But I want to know the portion that this homology, the results of the components from homology, like what portion of the data set, like, are they contributing to? And then while you were ending towards the conclusion, you talked about the performance in relation to homology groups, I think. And I want to connect the two, like the percentage of this created by the H3 within the CDR group. Like, which percentage are we talking about that comes from homology when it comes to the data set?
So all of our examples we run through modeler to generate a homology structure, and this is partly to reduce the biases of individual like crystal structures that are going into the model. And this sort of helps standardize all the examples. So the training data set is sort of uniform. I'm not sure how that relates to the CDR regions because the flexibility of these CDR regions don't change too dramatically when we do the homology models, just because these aren't as flexible, and modeler is really only replicating what the PDB template that you give it as. So the sequence identity here that we do the analysis on is coming directly from the sequence of the antibody that we're looking at. So this is sort of irrelevant of crystal structure versus the homology model, if that kind of answers your question. Yeah, go ahead, Sid.
Yeah, okay, maybe go back and show the homology model example. Right. So the idea here is that we have a single crystal structure for an antibody-antigen complex that's publicly available, but these are rigid bodies, right? A crystal structure is just a static representation of this protein complex. So we throw it through modeler to give us that ensemble, some little bit of flexibility. Some of these loops might be in different places for a given example. So for each, so we sort of expanded our data set, right? So we took a crystal structure and we made 10 versions of that crystal structure using homology modeling to give us structural homology. But then the sequence homology is the data leak issue. So in the field, there is a systematic problem of splitting the training, validation, testing sets. And if you look for structural and sequence homology, you'll get a better split. And I think many of the authors or groups are not doing that. And that's the distinction. So there's homology modeling for the structural ensemble to give us a better sampling in the model input. And then there's the homology filtering to help split our sequence-based splitting of our data set to give a better give them a more fair training and validation set instead of just inflating our generalization numbers by having data leak.
Okay.
So like the idea is that a multiple sequences may map to a single structure, right? Like you may have a varying amino acid sequence that gives you a type of interface, but people don't generally filter for that in the field. So you'll have a sequence that essentially looks or a structure that looks unique, but it is it's overrepresented in other examples.
Thank you. I think that answers the next question I had about data leakage. And I think that's great. So my next question is about really you did something that is, I don't know, am I audible enough? Hello?
Sorry, yes.
Okay, good. So my next question is something about the structural equivariancy. And you really revolutionized the CNN, because I'm one of the people who think probably the CNN has reached its limit, but seeing something new out of it is really revolutionary, and the way you used it. So one would wonder, like, because we can think about like augmenting like rotation and like feed it maybe as an input to the CNN. So what could be the practical advantage for equivariancy, like that is given non-equivariant approaches that where you could do rotation augmentation? And I feel like, just maybe you could add on this, I feel like the complexity is higher when you go for SE equivariancy rather than the rotation augmentation. So what's the trade-off?
Right. So I guess we didn't show this, but neither in the 2D or the 3D example, but if we wanted to solve this problem without equivariance, we'd have to, like you said, rotationally augment our input, right? Because our model wouldn't understand that this interface is the same as this interface, just rotated, right? That's something that the model will not know. So I think that is like, whenever you have to sample, it becomes it's a sampling rabbit hole. Like, when do you stop sampling? When is it good enough? So you can avoid all that sampling if your filters or kernels in the model are just aware that that the given interface is the same whether it's oriented differently. So that's one thing. But in terms of like optimization, so like trade-off, like would I rather just do a rotational ensemble input or just equivariance? I think the equivariance networks are, yes, they have increased complexity compared to the vanilla CNN or 3D CNN, but it's not that much more complex. It's just more weights, more filters. So you have, instead of this regular CNN has a static set of filters that you update weights in. This comes with a pre-initialized set of filters that have the symmetries encoded in them. So it's not that much more. And from our own benchmarking, it's still quite fast. The biggest actual bottleneck is the FFT, because we have to increase our grid size. There's something called Nyquist theorem, where if you want to study signal on a surface or in a space, you should pad the space to be twice what you need. So FFT is actually the bottleneck of our model.
Yeah, so the SE3Bind model is actually fairly simple in terms of parameters. I don't have the number off the top of my head, but it's around 12,000 or 13,000 parameters total. So it's a very fairly shallow and simpler model. So it's not as complex as you might think. Just we have the equivariance or SE3 added to it. So.
Wow. Thank you. Someone is asking, thank you for the talk and great work. Are you open to testing the model on an African dataset? I don't know how African datasets would look like in that context, but do you think it would give the same results? Yeah.
So I think you could train the model on a given data set. So for example, we shape the model for protein-protein interaction, right? Based on the 2D going to 3D. But Shriya decided to go to the antibody-antigen problem, right? That class of molecule. So in that sense, if you have a data set, if you have a set of protein structures, maybe you have crystal structure available, or maybe you just find your genes of interest and you generate a bunch of AlphaFold predictive structures. We could use these models to sort of screen interaction. We could maybe do like high-level screening of your generated structures and tell you what might be interacting. That's like that's how I would apply it.
I think he asks an intuitive question that now I come to think about it. When you think about, of course, African datasets, I think in this field, if they exist, what I know, like people like Chan Zuckerberg Foundation, they have done a lot of data collection, but I don't know whether the data set will look different, but if it's different, that means you're likely to face some sort of distributional shift within the model. So hopefully the model can still handle that shift. But yeah, I think he's asking something worth thinking about.
Sure.
Yeah, so this model was specifically for antibody-antigen, but I'm also working on another model that's more general and protein-protein prediction. So maybe that's something that could be fine-tuned further to general protein data sets. But I think this particular model, since it was trained and validated on just antibody-antigen, it would work well with the new antibody complex data instead of regular protein-protein interactions.
Finally, from my end, to Shriya and Sid. I've seen a lot of work in protein-protein interaction affinity prediction, but yes, we have, I think, discussed work in antibody-antigen affinity prediction. And I think from my judgment, antibody-antigen prediction seems harder than protein-protein affinity prediction. And I just from you to learn from you whether that is true, and if it is true, why the community? And even when you look at the work, a lot of work is focusing on protein affinity prediction, which makes me think like, yes, it is true, it is hard, but why? What makes it hard?
So currently there's a data limitation, right? So we don't have a large abundance of the binding affinity for antibody-antigen complexes. There's plenty of data for, regular protein-protein interactions. There's binding affinities, but for antibodies, I think just the data set is so limited that makes it really hard to train a robust generalizable model. And I think just the way these CDR regions evolve so quickly that it's really hard to sort of maintain a database of these, fast evolving CDR regions, changing CDR regions. And I think that adds to the structural complexity that we see. But I think the real issue is just limited data for training, because there's only about I think 200 actual unique complexes of antibodies that we use.
Okay. So it's mainly a data scarcity problem.
Yeah, exactly.
Okay. I think that's all from my end. I don't know whether there is anyone else in the audience that wants to add something. But if not,
I could add something. These models, this is just so the SE3 equivariant CNN. You can imagine that this is not just the end-all solution. This is just a component in an ecosystem of models. So you could have a workflow where you generate a bunch of synthetic structures using AlphaFold, and then you would screen them high level quickly with the ConvNet, and you isolate regions, and then you can go do refinement and do other things. So if for the future of these kind of models, I would use a variation of them based on a specific task. What are you interested in? How can you use what's available? So I would use a language model to generate my structures. I would use a vision model to find regions of interest and refine it from there. Potentially use a flow matching model to take our vector features and try to build a essentially a vector field for a correction of how to improve our structures, potentially things like that. So that would be my takeaway for this work and what we would do in the future.
Thank you. Thank you, Sid and Shreya. And I really want to look at the source code. I've been looking at the paper and I've realized there is a GitHub repo for the project.
Yes.
And I always encourage people, even just looking at the code, you don't have to understand it straight away, but just go to that repo and look through. You never know what you could obtain from there. So I want to thank you for taking off time and really share this wonderful work with us, to Shreya and Sid, and to your professor that really brought me in touch with you guys. Thank you very much. Thank you very much indeed.
Yeah, thank you so much for the
thank you.
invitation. Yeah.
Yes, and thank you to the members. Until next week.